Okay. So as I said, this section would center on the biological psychology aspects, just to deal with how certain behaviors could be explained using the biological dimension. So with regards to the biological dimensions, in explaining any behavior using the biological dimension, these are the core areas we tackle in any aspect, any clinician. These are the core areas they tackle. So it means you need to find out the neurochemical aspect. When you say the neurochemical, we are dealing with certain chemicals or neurotransmitters, especially within the nervous system, that is really affecting that kind of behavior. So physicians would explore that. Also, physicians would explore the neurophysiology, which is the various functions or reactions going on in the nervous system, which we will delve more into it. Then the next thing is to also tackle the neuroanatomy, how the various parts of the brain and the spinal cord works and all that. Then the endocrine system, which is how hormones could also affect behavior. Then the last thing is hereditary. So in terms of heredity, we're dealing with how genes in that high aspect also affect human behavior. But today's discussion will focus on the first three: neurochemical, neurophysiology, and then neuroanatomy. Today's discussion will center on these aspects. Okay. So right from, in terms of biological explanations, usually to explain these behaviors, we use these areas. We have several of them, but I decided to focus on these three main areas. We have the physiological explanation, ontogenic explanation, and evolutionary explanation. So those who always use the physiological explanation, they'll be the ones talking about how the brain or the spinal cord, its structure really affected a person's behavior. So they will be dealing with, if you want to explain any behavior, they'll be using the brain and the spinal cord and certain chemicals within the brain and the spinal cord, how it influenced the person's behavior. So for instance, if someone wants to explain why males are more aggressive than females, the person would then start from the fact that, when it comes to males, they have high levels of testosterone, which really makes them more aggressive than females. So they'll be using those chemicals within the various systems in the brain or the body to explain why this person is behaving the way he or she is behaving. Whereas those with ontogenic explanation, they wouldn't only focus on the physiological reactions going on within the body, but also examine how even people learn certain behaviors in their environment. So using the same aggression tendency, someone can say, oh, yes, it's true that males have high testosterone, but children, when they are exposed to certain, especially when male children are exposed to certain violent movies and everything, they learn more of these aggressive tendencies too, which react with their testosterone level to make them more aggressive, especially when they have one or two issues with people. So those with ontogenic explanation, they wouldn't only focus on the biological dimension, that's the physiological aspect, but would also factor in how the environment, like the child observing certain behaviors in the environment, like watching aggressive movies, action movies, and so on, how it could also interact with their biological dimension and lead to that kind of behavior. Then those with evolutionary explanation would trace our behaviors right from our ancestry And I know some of you have come across Homo sapiens, man apes and Homo sapiens. I think last week we delved in it and all that. That I said at first, humans were, when you check our physique and everything, we're kind of closer to like apes, chimpanzees, and everything. So during that Stone Age period, for instance, people normally use aggression as a way to flee out of something that they fear most. So someone who wants to explain why, especially males, are more aggressive than females will say, oh, right from ancient times, males are the ones who normally do the hunting and gathering. And they have built these instinctual drives that's being aggressive right from the hunting and gathering period as a way to flee from maybe wild animals. So they have these things in them. They inherited these things from their ancestors, and it has been passed on from generation to generation. So it is something which is part and parcel of what? Of males. So this is how they try to explain these biological explanations, and all these things falls under biological explanations, dimensions. Okay, so please, if you have a question, kindly use the emoji to raise up your hand. Then I would give you opportunity to ask your question. So I'm still moving on. All right. So in this discussion, we're dealing with the nervous system in itself. And I think it may be in anatomy or something you've been exposed to this. So whatever being the case, we'll still tackle it. So with the nervous system, it can be divided into two main parts. So we have the central nervous system and the peripheral nervous system. So central means it is in the middle. So when you check your body, you can use your body as an example. You can check that there are two main structures within your body that are more or less like in the midline or falls within the center. When you check the position of your head, it is more or less like position at the center of your body, which is the brain, where you can find the brain. Then when you check your spinal cord too, you can see that it divides your body into two equal halves. So it means that the central nervous system consists of the brain and the spinal cord. Then the other structures are usually referred to as the peripheral nervous system. So your legs, hands, stomach walls, all the other structures are then referred to as peripheral. When you say peripheral, it means the outer part. So the other structures that falls outside the central nervous system is referred to as the peripheral nervous system. Then within the peripheral nervous system, we can further divide it into two, which is autonomic nervous system and somatic nervous system. So soma means body. So it means that within the outer layer of your bodies, the muscle tissues that we find, that's the somatic nervous system. These are the somatic nervous system. Then within these muscle tissues, you can see that there are some nerves connecting them. Those nerves are what we normally call them the autonomic nervous system. The autonomic nervous system, the nerves within the muscles and everything, which communicate and all that is what is referred to as autonomic nervous system. So with the autonomic nervous system, we can further divide it into two parts, which is sympathetic nervous system and parasympathetic nervous system. Parasympathetic nervous system. So these are the two main parts you should know of in that regard. All right. So when you say sympathetic and parasympathetic, what does it really mean? So let me move on so that you would appreciate it more. All right. So I know you've come across this term, fight and flight responses. Let me type this, fight. Oh, so fight and flight responses. I know you've come across this. So if you have any fair idea, you can raise up your hand, then I would allow you to talk. If you have any clue about what the fight flight response is all about. Or even others even add fight, flight, or freeze responses. Any clue? All right, Leticia, please go ahead. You can unmute your mic and speak. Okay, so I think it's by mistake. Let me go ahead. So when it comes to the fight, flight responses, normally when people experience something that they are afraid of, they fear most. So for instance, let's say we have people who are afraid of cockroaches. When they see cockroaches, they'll be what? They'll be, you see them screaming, then they will run. Or when maybe they see a snake, you see the person was screaming, then running. So the person running, that means it is an automatic response that comes because the person has seen something he or she is afraid of. He or she doesn't have the necessary resources to fight it. So the person flees. So the flight response is the person what's running away from the thing that they are afraid of. Then the fight response is when the person have necessary resources. Maybe the person have seen that, okay, there's a stick, I can fight this thing I'm afraid of or I fear most. Pick the stick and hit the snake with a stick, then it means that there's a fight response. So it means during times whereby someone is afraid of something or there's anxiety, these are the key things that happen. Either you fight off or flee. Others, they wouldn't fight or flee, but the moment they see the thing, they can't even run away or fight. You see them what? Stand still. So that's the freeze aspect of it, which sometimes happens. So the person going through these things, it means that during that period, the sympathetic nervous system is really, really active. And physiologically, certain things are going on within the person's body. You can see that when the person even screams, you could see that the person's heart will be beating very fast. It's because of the sympathetic nervous system. So during times of fight, flight responses, physiologically, these are the things the person go through. That's what I've indicated here. All these things happen. Your pupil, which is the black eye, will dilate. When you say something will dilate, it will become large. Then the person can't secrete a lot of saliva, so saliva secretion will be low. Then what happens is that the heart will be beating faster. That's why they've indicated accelerate heartbeat. Another term you might come across is there is palpitation. Palpitation. It means the same thing. The heart will be beating very fast during that time. Then in terms of your gastrointestinal organs, your gastrointestinal organs. So that means your stomach walls, all the structures within your abdomen, they wouldn't be highly activated. It will be inhibited. All those structures inside will be inhibited. Yeah, so these are some of the reactions they go through. So all these things people go through during times of fight, flight responses because of the sympathetic nervous system being activated. So you can see that when a person goes through the fight, flight responses, with time, whatever reactions the person experiences slows down so that the person will come to a normal state. So the person's coming to a normal state whereby those reactions are slowed down, it means that during that time, the parasympathetic nervous system will be activated. So initially, if during the time of fight, flight responses, the pupil dilated, with time, the pupil will begin to constrict. You remember I said when it dilates, it means it becomes large. So with time, it begins to become smaller. That's what I mean by it constricts so that it will come to a normal state, the black eye. Then your heart, that was initially beating faster begins to slow down because the parasympathetic nervous system has been activated. So it means the parasympathetic nervous system does the opposite of what the sympathetic nervous system does. So when you check these things, you can see that it is direct opposite of the sympathetic. With the sympathetic, we said that people would dilate, but with the para, it constricts. And this one, we said there's acceleration of heartbeat, but here the heartbeat slows down. So that's how these reactions takes place in that regard. Okay. As I said, I'm still continuing. If you have a question, you can use the emoji to raise up your hand. Then I would pause and give you opportunity to ask your question. All right. So in terms of the nervous system, whether it is central nervous system or peripheral nervous system, it has two main cells. It has two main cells. We have neurons and glia cells. So generally, this is what a neuron does. It receives and also information and also transmits it to other neurons or cells. Then glia cells, it performs several functions, but I've indicated some few of them. So let's go through what neurons are, the structure of a neuron. Generally, this is how a neuron looks like. So within especially the brain, this is how you will see the neuron to be like. This is the structure of the neuron. So it has several parts. So the branches that you see is normally referred to as the dendrites. And within the dendrites, in a microscopic manner, it has some spines known as the dendritic spines. Within the dendrite, it has these microscopic spines known as dendritic spines. And the role of the dendrite is to receive incoming information. So for a neuron to receive any information, the information needs to first sit on the dendrite. So the dendrite will be the one, since it is a branch, it will be the one to receive information coming to the neuron. Then the next structure within the neuron is the cell body, also known as soma. So the cell body is also referred to as soma. So this area. So after the information, several information has been received by these dendrites. The various incoming information would come to the cell body, whereby it would sum these information together as one unit. So we normally say that the cell body integrates these incoming information as a single unit. That's the function of the cell body. That's the function of the cell body. And the cell body has several structures. So I think back in SHS and JHS, you can see that most of your teachers will say, oh, we have two main types of cell, animal cell and plant cell. Then they'll tell you, oh, animal cell or plant cell, they have these structures. Ribosome, cytoplasm, mitochondria, ribosome. You've come across some of these things. Mitochondria, cytoplasm, and what have you. All these structures earlier, back in SHS and JHS, you were exposed to, you find them within the cell body. It is the cell body that contains these structures, which helps to integrate the information coming, the nucleus and so on. All right. So after the cell body has integrated the information needs to travel along this long thing. It needs to travel along this long thing. And we call this lumbar axon. We call it axon. And even within the axon, we normally say there's this part, which is the first part of the axon. It's normally referred to as axon hillock. Axon hillock. And the axon hillock, we normally say it is the first area. So you can jot this somewhere. It is the first area in the axon where action potential begins. So we'll delve more into what action potential is, but for the meantime, know that axon hillock, it is the first area in the axon where action potential begins. Okay. Then it means that when it comes to the axon in itself, generally, impulses or information travel along the axon. That's the function of the axon. Impulses or information travel along the axon. So the axon allows information to travel along it. Then the last thing which forms part of the neuron is the axon terminal. Axon terminal, which is more or less like the root of the axon. And it is sometimes referred to as the presynaptic terminal. The presynaptic terminal. Okay. So the presynaptic terminal, it is the area where chemicals are transferred to other parts of the neuron. It is the area where chemicals are transferred to another neuron. So we'll delve more into it later. So within a normal, this structure is more or less like a normal axon. And this structure too is an axon, but you can see that there has been some distinction. There's a difference between this axon and our axon A. The only distinction is that with this axon, it has this thing surrounding it. It has this thing surrounding it. And there's a reason as to why it has this thing surrounding it. Some axons, not every axon has this thing surrounding it. And this thing surrounding it, it contains both fats and proteins. It is composed of fats and proteins. And when these fatty substances surround an axon, we call that fatty substance myelin sheet. Myelin sheet. Then you could see that within this junction, there's no fatty substance surrounding it. When you check well, let me erase this so that you get it well. So you can see that within the middle line, there's no fatty substance surrounding around the side. And that area is normally referred to as nose of Ranvier. Nose of Ranvier, or Ranvier, any way that you pronounce it. So generally, we call this entire structure a myelinated axon because myelin sheet surrounds it. So we normally call it myelinated axon. And within a myelinated axon, so you should pay attention well, within a myelinated axon, action potential takes place within the nose of Ranvier. So I said we would delve more into action potential. But one thing you should know is that within any myelinated axon, action potential takes place around the side, the nose of Ranvier, this area, whereby there's no fatty substance surrounding it. That's where action potential takes place, nose of Ranvier. All right. So within the brain, for instance, we can have several, the axon have billions of cells within them, especially in the brain. So it's not like, okay, within the brain, we have 1 or 2 axons. We have billion axons, and they communicate with each other in terms of that. All right, so Miss Anne Gabra, please, your hand is up.
Yes, please, sir. Good morning.
Morning.
Sir, please, the last statement you just made along the axon where there is no fat and then protein, the junction was showing us.
Okay.
You said something that's I didn't get it.
Okay, I'll record on it. Don't worry.
Okay, thank you.
Okay, so I said within a myelinated axon, action potential takes place at the nose of Rambier within a myelinated axon. So you know that, okay, when I say myelinated axon, it means that the myelin sheet is surrounding that axon. So we are saying that within that myelinated axon, action potential takes place within that junction where there's no fatty substance surrounding it. That's nose of Rambier. That's where action potential takes place. Okay. So I'm continuing. So I think I've already explained these things. So within the myelin sheet again, the reason as to why some neurons have these myelin sheet is that the myelin sheet, you remember I said information or impulses travels along a normal axon. Impulses travel along this axon. But some axons have myelin sheet. Why? Because the myelin sheet allows the information to travel faster. So some axons that have myelin sheet, the essence is that it allows the impulses or information coming or traveling along the axon to travel at a faster pace than a normal axon with no myelin sheet, which we call it unmyelinated axon. So a normal neuron with no myelin sheet surrounding it. With that, the way manner in which information is traveled along the axon is way slower than if a myelin sheet surrounds that axon. Okay, so that's one of the core function of the myelin sheet within an axon. All right, so in terms of neurons, we have 3 main types. So irrespective of the type of neuron, the key thing is that in every neuron, you will find these structures. The axon, dendrite, soma, and terminal button. Irrespective of the type, whether it's a sensory neuron, motor, or interneuron, you have these structures forming it. All right, Matilda.
So please, if I understand what you said, that you said axon hillock is the first area where axon potential begins. And you also said with the nose of Rambia, is where action potential takes place. Is there a difference?
So if you've really paid attention, where I said with axon hillock, that's where action potential begins. That's the axon or the neuron with no myelin sheet surrounding it. And so it means that when there's no myelin sheet surrounding it, the first area where action potential begins is the nose of Ranvier. Then afterwards, action potential would also run through the entire axon. So it means within an axon, a normal axon, or within a neuron, action potential goes through within the axon. But I said axon hillock because I said axon hillock is part of an axon, and that's the first area within the axon where action potential starts. But with with a myelinated axon, always action potential will start from, or would always happen within areas in the axon where there's no myelin sheath surrounding it. So it means when you check this area, for instance, since there's no myelin sheath surrounding it, and this one is also part of a neuron, action potential will take place. This side too, action potential will take place. So within an unmyelinated axon, you have several action potentials taking place, multiple action potential taking place. So let's say this is a myelin sheath we are using to surrounding this neuron. There's also another myelin sheath, and we have another myelin sheath. So it means that within this neuron, we will have action potential taking place here, action potential taking place here, action potential taking place, action potential taking place, action potential taking place. So you within a myelinated axon, depending on how the myelin sheath surrounds that axon, you have multiple action potential within that sort of axon or neuron. That's how it works, because every area whereby there's no myelin sheath, the moment the information gets there, action potential or will take place. That's how it works. All right. Okay, I think, Matilda, I've answered your question, so I'm lowering your hand. There's this person, iPhone. You've indicated yes, I think your name, you've raised up your hand, so please go ahead.
Sir, please.
Okay.
If I
could barely hear you. It seems your network is kind of poor.
Does it mean that with the myelinated, sir, please, does it mean that action potential takes place in the node of Ranvier only?
That's what I've been echoing on right from the start.
Yeah.
That within a myelinated axon, action potential always begins within the nodes of Ranvier because there's no myelin sheet surrounding it.
Oh, thank you.
All right. Okay, so I'm moving on. All right, so we have different types of neurons, and as I earlier opined, irrespective of the type of neuron, always there would be these structures. That's dendrite, soma, terminal button, and axon. They have these structures. So the first type of neuron we have is sensory neuron. Sensory neuron, also, or sometimes referred to as afferent neuron. So it is a neuron that usually picks information from the environment and then send it to the central nervous system. And right now, you know the main composite of the central nervous system. That's the brain and the spinal cord. So that's why I have indicated the peripheral nervous system. So it means that when we pick information from the environment, like you want to see something, the sensory neuron will pick the information from where? From the eyes, because the eyes is part of the peripheral nervous system. So the sensory neuron will pick information from the eyes and then take it to where? To the brain, which is the central nervous system for interpretation. So that's why I've indicated it carries information from the peripheral nervous system to the central nervous system. It's the same as me saying it carries information from the environment to the brain or spinal cord, because they fall as part of the central nervous system. All right. Then the motor neuron is also referred to as efferent neuron. This time around, it would carry information from the central nervous system that's either the brain or the spinal cord, to the peripheral nervous system, which is, say, the eyes. So let me give you a mechanism. And these two neurons, the sensory neuron and the motor neuron, they work jointly together. So for instance, for you to see that, oh, this is what we've projected, or for you to say that, oh, this is a red blanket, it means that your sensory neuron will pick the information from the environment. That's from the eyes. Then it will take to where? To the brain for interpretation. Then the brain will let you perceive that, oh, so there's these things are more red. So the information needs to further come back to your eyes for you to see well and say, okay, this is what, a red blanket. And for the information to come back to your eyes, the motor neuron will be the one that will pick the information interpreted by the brain. So the motor neuron will pick information interpreted by the brain to your eyes. So that's why we are saying it carries information from where? From the central nervous system, that's either the brain or the spinal cord, to the peripheral nervous system, whereby I said the eyes is also part of the peripheral nervous system. All right. Then the next one is interneuron. Interneuron is sometimes referred to as relay neuron. And its function is that it integrates information between the sensory neuron and the motor neuron. So a typical example is women who normally cook. So let's say in most situations, when you want to pick, maybe you are cooking, you realize that, okay, the meal that you were preparing, or you've exhausted the time and everything, so it's enough for you to pick it from the hot saucepan. Sorry, from the hot plate. So unfortunately for you, you forgot to use what a napkin. It means that you feel some pain, right? So you feeling the pain, it means that your sensory neuron will be the one that will pick the information to your brain and say, oh, the person picked or touched the hot plate. So there's a burning sensation. Then your motor neuron will pick that burning sensation and say, oh, since there's a burning sensation, the motor neuron will pick the information from the brain and take it to your hand and say, oh, withdraw your hand from where? From the hot plate. So you withdrawing your hand, it means that your motor neuron is active and the information is being sent to your brain that for you to know that, oh, it is a hot plate that you've touched. That means your sensory neuron is the one doing that. But in a situations whereby the same thing, you are cooking, this time around, you use a napkin to pick it. That means your interneuron will be activated. And that's how it works. First, the same information will be carried to the brain for you to perceive that, oh, the thing that you've picked is really hot. Then your interneuron will then communicate and say, oh, it is hot, all right, but the person is using what? A napkin. So the motor neuron shouldn't communicate to your hand for you to withdraw your hand because you are using what? A napkin. So that's what we are saying. The interneuron communicates information between the sensory neuron and the motor neuron. So long as you are using a napkin, then it means the interneuron will signal to the motor neuron that, oh, don't take your hand off from where? From the saucepan because you are using a napkin. So that's how the mechanism works. Okay. So you remember I said we have two types of cells when it comes to the nervous system. We are done with neurons. We are moving on with Glia cells. Glia cells. All right. So Glia cells are sometimes referred to as neuroglia. Glia cells are sometimes referred to as neuroglia. Neuroglia. Okay. So the term glia cells, as I've indicated, came from a Greek word known as glue. So when you compare glia cells with the neurons, glia cells have they are more in the brain and the spinal cord than neurons, and they perform several functions. We have different types of it. So these are the main types of glia cells you should know of. These are the main types. And I've given you a pictorial view so that you would also have a fair idea about the various glia cells and all that. Okay, so let's go through them one after the other to find out the functions of each of the glia cells. So the first one is the astrocyte. Astrocyte. And astrocytes, they have this star shape. This shape is more or less like a star. The same thing applies to this. This is also an astrocyte. It has this star shape. And one of the main functions of the astrocyte is that it provides structural support for other cells and neurons. So when you check, especially this type of astrocyte, this purple astrocyte, you could see that it is also preventing this capillary, this reddish capillary, from what? Falling. By what? By more or less like entangling this capillary so that the capillary wouldn't fall. So that's what I mean by it provides structural support for other cells and neurons. When you check this neuron too, this is a neuron. That's why we have this myelin sheet surrounding it. When you check this neuron, you can see that the astrocyte again has entangled it, preventing it from also what? Falling. So it is providing a structural support. So you can see that when it comes to this neuron, for instance, we have several myelin sheets. This is the myelin sheet that I was talking about. And within it, we have this space. That's the nose of Radialglia. So that means several action potential will take place within this myelinated neuron. Yeah. Okay. Then apart from the astrocyte performing structural support, it also removes certain waste materials or debris from the brain and the spinal cord, or from the nervous system. So usually when some cells die within the nervous system, the astrocyte will be the one that would remove those dead cells so that it wouldn't cause certain infections or damages to the brain. Apart from that, the astrocytes, again, sometimes provide nutrients. They provide nutrients. Some nutrients like fatty acids, then protein, oxygen, and what have you, to other cells. So basically, these are some of the functions of astrocytes. All right. So the next one is Radialglia. Radialglia. So Radialglia, that's these are all Radialglia that have provided, and this is a Radialglia that has entangled a structure. I think it might be, yes, it has entangled it. So Radialglia, its function is that during the prenatal stage, we call a stage known as the embryonic development. It allows neurons to migrate, especially when it comes to axons. It allows these axons to migrate to other parts of the bodies. So basically, that's its function. During embryonic development, whereby the fetus is developing, whereby the brain is developing, then Radialglia performs a key role. It allows especially these neurons to develop within the brain and other nervous system. That's the core function of the Radialglia. All right. Then we have the ependymal cells. Ependymal cells. Ependymal cells. So you can find the ependymal cells around the meninges. So you can read more about where to locate the meninges. It is when you open the skull, we have 3 layers that protect the brain, and we call them meninges. And within those areas, we have ependymal cells. And the ependymal cells helps in producing a fluid known as cerebrospinal fluid. Cerebrospinal fluid. And the key function of CSF, which is cerebrospinal fluid, is that it cushions the brain. It also nourishes the brain. So before you get to the brain, there's a fluid surrounding it, and that fluid is what is referred to as cerebrospinal fluid. Yeah, so for those fluids to be produced, the ependymal cells are the ones that normally help in the production of it. Okay. Then the next one is microglia. Microglia. So when it comes to microglia, its main function is that it, to a large extent, helps destroy pathogens, bacteria, viruses, fungi, and other microorganisms. That's the main function. So it not only removes debris, like what the astrocyte does, but its core function is that it helps destroy these pathogens like bacteria, fungi, viruses, and what have you within the cells or the nervous system. So we normally say that it is more or less like the immune system of the nervous system. It is the immune system. So for us to say that, okay, your body is immune to A, B, C, and D, it means that these microglia will be activated to do that function. All right. So in most situations, in books, instead of they saying, oh, these microglia helps destroy fungi and other pathogens, they will say it serves as a phagocyte. So there's a term you might see or come across. It serves as a phagocyte. That means the same thing. Or so in terms of it helps in removing these pathogens, we call that process phagocytosis. Phagocytosis. That's the process. All right. So if you have a question, feel free to draw my attention by using the emoji. Okay. Then we have these structures. That's the first one that we have is the Schwann cell, which is also part of the Glia cell. So Schwann cells and this structure, oligodendrocytes, it is the same structure. It is the same structure, just that when it is found within a particular aspect, it has different name. It has different name. So when I say it is the same structure, let me show the myelin sheet that we've been talking about. It means that it is either a Schwann cell or oligodendrocyte, the myelin sheet. So when a neuron has a myelin sheet surrounding it, it is either called a Schwann cell or oligodendrocyte. So it means when a myelin sheet, for instance, is found within the peripheral nervous system, when a myelin sheet is found within, say, the muscles, so this is like a muscle, whereby you find this myelin sheet within the neuron of a muscle. So it means that we will call this entire structure or neuron Schwann cell, because the myelin sheet was found where? Within the peripheral nervous system, which is the muscle. in a situation whereby the myelin sheet this time around is found within, say, the brain or the spinal cord, then we wouldn't call it myelin sheet at the moment, but rather it will be called oligodendrocyte. Or sometimes in certain books, they will say oligodendroglia. It means the same thing. In some books, they will say oligodendroglia. It means the same thing. It means there's a myelin sheet surrounding a neuron, which can be found in either the brain or the spinal cord. Okay. So that'll be all when it comes to different types of cells, which is neurons or glia cells. So please, any question? Any question or clarification? All right. So I'm continuing. So where's the eraser? Okay, so I'm continuing. So the next thing that we'll be tackling is nerve impulse. Nerve impulse. So you remember I said within a neuron, for instance, action potential. Okay, Madam Ya Sewa, your hand is up. Please go ahead. Please go ahead.
Good afternoon.
Good afternoon.
Please, during our diploma, ganglia, is it part of what we are learning now?
I'm confused here. You said during your diploma what?
Yes, I heard something like ganglia, and the name sounds like similar to what we learned before this. The Glia cells.
I haven't mentioned any ganglia. The only glia cells I mentioned was oligodendroglia, sometimes also referred to as oligodendrocytes. Ganglions are different cells altogether. Sometimes you find these ganglions, especially in your eyes. Whenever you are treating eyes, you can find these ganglions there. And that's not my focus.
Okay, thank you, sir.
All right. Okay, so the next thing is nerve impulse. Nerve impulse. So with nerve impulse, you remember I said when it comes to neurons, in most situations, action potential, when I say action potential, it means that impulses traveling or information traveling within the axon. That means action potential has taken place. So action potential takes place within the axon. So we want to find out what really happens when action potential is taking place in an axon. What are some of the things that goes on in that sort of axon? So that's what I mean by nerve impulse. So when you say nerve, it means information is traveling within that sort of axon. So the information traveling within the axon, what are the key things happening in that sort of axon? So when the cell is at rest, that's when it is in a resting potential. So for us to say that the cell is in a resting potential, that means there's no information traveling within that sort of axon. That's what I mean by the cell is in a resting potential. It means nothing is happening in that sort of axon. So we are saying, ideally, when the cell is in a resting potential, there are certain ions, and these ionic distributions are what are different. All right, so let me use a pictorial view so that you get the concept well. So let me move on to this aspect. So usually we are saying when a cell is at rest and there's no movement, let's say we have cut the axon. There's an axon, and we have cut it to know what is really inside. So this is the inside walls. So when we cut an axon, the inside walls is sometimes referred to as intracellular cell. Intra, so intra means inside. So intracellular cell. Whereas the outer layer of that axon would be called extracellular cell. So we are saying that when we cut that axon, the inside cell, which is the intracellular cell, it has some ions. And the main ions you find when a cell is at rest, it is in a resting potential. The main ions you find is potassium, which is K+, then other proteins, which is A-, other proteins. So other proteins means it has several ions coming together as one. So it means within the inside cell, this layer, this area, within this area, you find a lot of potassium, which is K+, and A-, a lot, found often here. Then at the outer layer, you find a lot of sodium, Na+, sodium, and chlorine, Cl-, a lot at the outer layer. So what we are saying is that, especially when in chemistry, when you combine sodium and chlorine, you get what? Sodium chloride. And sodium chloride, when you taste it, it is very salty. So we are saying even when a cell is at rest and you taste the outer layer, you can see that the place is very salty. That's the essence, meaning that it has a lot of sodium and chlorine in there. Okay. So when a cell is at rest like that, that means no information traveling. And all of a sudden, information begins to travel. Certain things happens for that to happen. It means that the moment information begins to travel within that cell, these ions, their distribution changes. These ions, wherever we are saying sodium and chlorine are found a lot outside the cell, then potassium and other proteins are found a lot inside the cell. It begins to change. That distribution begins to change, leading to information traveling within that sort of axon. And that's what today's discussion is all about. So it means for information to travel along that sort of axon, it has to do with two main forces. Two main forces are at work for information to travel along the axon, and they are electrical gradient and concentration gradient. So these are the two main forces at work: electrical gradient and concentration gradient. All right. So let's find out, and before I talk about the electrical gradient and concentration gradient, one thing you should also notice that when it comes to these ions sodium, potassium, chlorine, and other proteins, they all have their unique gates, which solely allows that ion to pass through. So when you check this green gate, for instance, that's sodium gate. So this green gate only allows sodium to pass through, and no other ion will pass through that gate. When you check this red gate, it allows only potassium to pass through and no other ion. So we normally say that usually the cell is selectively permeable. What it implies is that every ion has its unique gate, which allows only that ion to pass through. So that's why we are saying it is selectively permeable. Okay. So moving back to the two forces. So let's find out what really happens in terms of the concentration gradient and electrical gradient. So for information to travel along the axon, first of all, the information needs to, one of the main force has to do with laws of diffusion. And as you already know about diffusion, so what is diffusion? That's my question to the class. What is diffusion? Any clue? What is diffusion? I think you did osmosis and diffusion. So what is diffusion? Okay. Sorry, I could barely hear you.
I said is the concentration to a region of lower concentration until the molecules are dispersed.
Okay. I couldn't hear it well, but I think I get where you are coming from. Okay, Joyce. Joyce, your hand is up. Okay.
So I think is the movement of molecule from a region of a higher concentration to a lower concentration.
All right. Yeah, you're right. So you're right. So diffusion is what is the movement of molecules from a region of a higher concentration to a region of a lower concentration. Yeah. So it means by loss of diffusion, let me draw the cut. So let's say this is the axon that we've cut into half. And we are saying this area has a lot of sodium and chlorine, a lot. The outer layer. Then the inner layer has a lot of K+. And A-. So when it is at rest, this is how it's like. And one thing about the cell being in a resting potential, we normally say that since it is at rest, it is negatively charged. I forgot to mention that it is negatively charged. So it means that if it is negatively charged, it means it has a lot of A- in there. So when you use an oscilloscope to measure the charge ions, we normally say it has about -70 millivolts. So when the cell it is at rest, it is in a resting potential, it is negatively charged, meaning that it has about -70 millivolts. of charged ions. That's what it basically means. Okay, so for it to move from a resting potential to an action potential, as I said, loss of diffusion first needs to take place. So it means the sodium ions, which is a lot, it is positive and also a lot or highly concentrated at the outer part, which is the extracellular cell. By loss of diffusion, needs to go where? It will go to the intracellular cell, because by diffusion, it needs to move from a region of a high concentration to a region of where? A lower concentration. Then again, by loss of diffusion, chlorine, which is highly concentrated outside, needs to go to the inner cell. Then K+, which is highly concentrated inside, needs to go to the lowly concentrated area, which is the outer layer. Then under proteins, which is also highly concentrated inside the cell, needs to go to the outer layer. But one thing about other proteins is that, you remember I said other proteins means it has several cells combined together, several ions combined together. It is really big and its gate is small, so it wouldn't get opportunity to move outside, although there will be pressure inside the cell that other proteins need to move out, but it wouldn't get opportunity to move out of the cell. So it means that by loss of diffusion, 3 ions are more implicated. Sodium and chlorine will move into the cell, then potassium will move out of the cell. So that's how the ionic distribution is like during period of concentration gradients. Okay, so please, any questions so far? Any question or clarification? So Matilda, your hand has been up for a long time. I don't know whether you still have any clarification. If no, then you can lower your hand.
No, please, I wanted to define the
All right. Okay, so remember I said we have 2 forces. So after the concentration gradient has been activated and done its job, let's see how the distribution is like within and inside the cell. So when it was at rest, we said potassium was a lot here and A- was also a lot here. And when it was at rest too, we said sodium was a lot here and chlorine a lot here. But the moment diffusion took place, some little sodium came to the inner cell. So you'll find some sodium here. Then you also find some chlorine here. Then during diffusion, some potassium also came to the side, although they are not a lot, but few of them came here. So it means this is a pictorial view of how, after diffusion, how the cell looks like. So right, the moment diffusion takes place and it does a job, then the next force that will happen is the electrical gradient. The electrical gradient. Someone is saying she can't see my screen. Then I think it might be coming from your end. So please adjust yourself. Or maybe your network is bad. Juliana, it's not from my end. It's from your end. So please adjust. All right. So this is how the concentration looks like. The distribution looks like, sorry. So with electrical gradients, let me throw your mind back to SHS and JHS. We usually say that in when they were treated, you were exposed to magnets. we normally say that opposite poles attracts and like poles repels. Opposite poles attracts and like poles repels. So since this is how the distribution is like after diffusion, sodium, which is positive, and it is highly concentrated outside, would be attracted by A minus, which is negative, because we said opposite poles attract. So it means sodium, which is positive, and it is also highly concentrated outside. Although some portions of it were able to move into the cell during diffusion, it is still highly concentrated outside. So since it is positive, it will be attracted by A minus, which is also highly concentrated inside the cell. So it means during electrical gradient, sodium will get opportunity to again move into the cell. So during electrical gradient, it is only sodium that will get opportunity to move into the cell. So it means, if you check very well, during concentration gradient, that's diffusion, sodium moved into the cell. Electrical gradient, sodium again had opportunity to move into the cell. So it means that if something is not done to sodium, it would flood the cell, the inside the cell, to flood it. It will make it a lot. So that will be the time the system has been mechanized in such a way that there's a pump. There's a pump known as the sodium-potassium pump. And its function is that this time around, after electrical gradient has taken place, the sodium-potassium pump will then pump out 3 sodium from the cell. Then the potassium, some portions of the potassium that was found at the outer layer, 2 will be pumped into the cell. That mechanism will be done. So when 3 sodium is pumped out, 2 potassium will be pumped inside. And it is done in such a way that it will put the cell, or it will bring the cell into what? Into a resting potential stage. A resting potential stage. All right. So that's how the nerve impulse happens. So please, any question? Any question? Up till now. Any question, please? Okay. If there are no questions, then I'm moving on. So I just drew a pictorial view in a graph form as to how it works. So you remember I said when a cell is in a resting potential, the volt is minus 70 millivolts. So that means the cell, this side means that the cell is at rest. There's no movement or information traveling within the cell or axon. The moment the cell or information travels within it, and for action potential to take place, the cell needs to cover up to this side. It needs to be positively charged. So for it to be positively charged, it means that sodium needs to move into the cell. So that's why you saw that during electrical gradient, sodium was moving because sodium is positively charged. During electrical gradient 2, sodium was rushing into the cell. So when it happens like that, then it means that the cell, that's when I say the cell, it means inside the cell. So inside the cell became positively charged. Then we can say action potential has taken place. So for action potential to take place, it means that the inside cell should be around plus 30 millivolts. The inside cell should be around plus 30 millivolts. It should move from minus 70 millivolts to plus 30 millivolts. So in certain books, they will write plus 30 MV. MV means millivolts. Then we can say action potential has taken place. And One key thing is that for action potential to take place, it is due to something known as a threshold. For action potential to take place, it is due to something known as threshold. So I'll explain what the threshold is. And usually in a cell, the threshold is minus 55 millivolts. So what we are saying is that in some cells or in some impulses, it wouldn't lead up to action potential. So for a cell to move into an action potential stage, it needs to cross a threshold. So let's find out what a threshold is. So that's what I've indicated, the point at which a neuron has received sufficient stimulation to trigger an impulse. So it means that when certain information is coming and it doesn't hit this cross mark, minus 55 millivolts, there's no way it would hit an action potential. There's no way to hit the plus 30. Some impulses, when it is within, let's say, minus 60 millivolts around this area, or even around minus 59 millivolts, it wouldn't what? It wouldn't get to an action potential because it hasn't really reached the threshold, which is minus 55, or it hasn't really hit the minus 55 millivolts. So there's no way it would what? It would lead to an action potential. So that'll be the time a principle known as the all or none principle comes in. So this explains the all or none principle, saying that for any stimulus or for any cell to reach an action potential, it needs to hit a threshold. If it doesn't hit the threshold, there's no way it will cause any impulse to happen or action potential to take place. So that's the all or none principle. Sometimes when you read certain books, they will use certain terms to confuse you about what the all or none principle is all about. But as I've simply explained, the principle that neurons only send an impulse. So when I say it only sends an impulse, it means when it only sends an action potential, or when it only neurons, when neurons, for a neuron to get to an action potential stage, it should hit the threshold, which is the minus 55 millivolts. All right, so I think this truly captures what the all or none principle is. You could see that this stimulus, for instance, led to what? Led to an information happened, but the information couldn't reach this threshold. So it means no action potential took place. This one too, although there was stimulation, it couldn't cross the threshold mark. So it didn't lead up to any action potential stage. But the moment this thing crosses the threshold, then it means that no matter what, it will cause an action potential to happen in that regard. All right. All right. So let's go through some key terms, terms in nerve impulse. So we have something known as repolarization. So it means the cell membrane returns to its original level of polarization. So this is how repolarization is. It means that after depolarization or action potential has taken place here, the cell is coming back to rest. That's what I mean by repolarization. It is coming back to its original state. But before it comes back to rest, which is minus 70 millivolts, it will first go to a particular stage. So you could see that in this stage, when you track it on the graph, this is more or less like minus 90 millivolts. So it means that during repolarization, the cell will further become negatively charged, too much charged. So it becomes excessively negatively charged. And when it happens like that, then we call that stage a hyperpolarized stage. So during repolarization, the cell first needs to come to a hyperpolarized stage, then later it will come back to its resting potential. It will come back to its resting potential. That's minus 70 millimeters. And during this period, during the hyperpolarized stage, we call a stage known as refractory period. Refractory period. So this is an assignment to you. Read more about refractory period. Refractory period. During that stage, the cell is in a refractory period stage. And we have two types of refractory period. I'll just mention them. Just find out what they really mean. We have absolute refractory period. Oh, let me move it to the side. Yeah. We have absolute refractory period and relative refractory period. So these are the two main types of refractory period that we have. Absolute refractory period and relative refractory period. So let me explain them so that it will be quite easier. So one thing about refractory period is that it lasts for about 400 milliseconds. So you can see that it is very fast. During the hyperpolarized stage, we are seeing that the cell moves to a refractory period. And this refractory period lasts for 400 milliseconds. That means less than half of a second. Less than half of a second. And we said we have two types. That's absolute. With the absolute, it starts from the first 200 milliseconds of refractory period. So the first 200 milliseconds of refractory period is referred to as absolute refractory period. So when it comes to this period or stage, we are saying no matter the intensity given to the stimulus, it wouldn't cause another action potential. So we are saying during refractory period, no matter the intensity given to the stimulus, it wouldn't cause another action potential to happen. Whereas with relative refractory period, we are saying it spans from the later 200 milliseconds to 400 milliseconds. So it means with relative refractory period, we are saying when the intensity of the stimulus is high, when the intensity of stimulation in the stimulus is high, when the intensity of stimulation in the stimulus is high, it will cause another action potential to happen. So I'm repeating, with relative refractory period, we are saying that when the intensity of the stimulus is high, it could evoke another action potential to happen. So a typical example of refractory period is when someone has numbness. I know you all know what numbness means. Once it will be inside T3T, let me say it in P. Inside T3T, that's numbness. If you really observe, when someone's hand is numb, within some period, when you touch the person, even when you pinch the person wouldn't feel it. So that's what the relative refractory period. When the place is numb and you try to pinch the person or touch the person and the person doesn't feel it, then that means that period is it's absolute refractory period. Because with absolute, we are saying when the intensity of the stimulation in the stimulus is even high or whatsoever, it wouldn't cause another action potential to happen. So it means during that period, when you touch the person and the person still doesn't feel it, we call that period absolute refractory period. But you could see that with time, when the numbness subsides or decreases a bit, then you pinch the person hard, and all of a sudden the person tells you, I can feel it, then it means we call it relative refractory period. Relative refractory period. Yeah, so that's the distinction between the two. So please, someone has been scribbling on my screen. So please, this is from doing this, I beg. All right. All right. So let me prevent anyone from scribbling. So please, I'll be with you. I'm trying to disable. Okay, I can't even find it. Let me continue. All right. So please, any question concerning this? Any question? All right. So the last thing that we'll be doing, then we'll be done for today. So after information has moved within the axon, that's nerve impulse have traveled along the axon, it needs to go to the terminal button. It needs to go to, you remember the root of the axon, the last base of the axon. You remember I said that's where chemicals are transferred to other neurons. So within the terminal button, there's a pictorial view of how. So this is a neuron. There's a red neuron. Let me put it in that fashion. Then we have this yellow thing, which is also a neuron. So within this area, there's a gap which we can't really see with our naked eyes, and that's the gap we have here. Within two neurons connecting each other, there's a space between them, but you wouldn't be able to see with your naked eyes. And that is where communication really happens. So this is like an axon. This is also an axon. And within this axon, this side is the terminal button, whereby after information has been traveled, it needs to go to the terminal button. Then the terminal button needs to release whatever information it was traveling along the axon to neuron B. This is neuron B. But you can see with neuron A and neuron B, there's a space in here. And we call that space synapse. We call that gap or space synapse. Or sometimes we call it synaptic cleft. So when you say cleft, it means there's a gap. Cleft means there's a space. So synaptic cleft. And there's a rationale as to why there's a gap between the two neurons. There's a reason as to why there's a gap between the two neurons. These gaps allows chemicals to be transferred to the other neuron. So let's find out how the mechanism works. So as I said, this is neuron A. When I draw, what's up? So this is neuron A, which is the terminal button. Sorry, this is neuron A. Then this structure is another neuron, neuron B, receiving the information from neuron A. So this side is the terminal button for neuron A. Then with neuron B, this side is the dendrites for that neuron B. You remember I said dendrites receive the information. So with neuron A, this long bar is the axon that we are referring to. So the axon is transporting something. When you say there's a nerve impulse or action potential, it means the axon is transporting something. So it is more or less like carrying this thing. This thing is what the axon is carrying or transporting. And this thing is usually called vesicles. 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 V-E-S-S-I-C-L-E-S. It is vesicles. And within the vesicles, you could see there's a brownish thing. Some like C's. If you check it very well, it's more or less like C's. And the vesicles also contains, or is also carrying a chemical known as a neurotransmitter. So it means that ideally, these axons are more or less like transporting neurotransmitters. So the moment this vesicle, let me erase everything. Yeah. So the moment this vesicle gets to the base of the terminal button, so when this vesicle gets to the base, when I say the base, this area, something interesting happens. The moment it gets to this side, then this green thing, which is calcium, this purple thing, which is calcium, would fuse with the vesicle and allow the vesicle to lose its identity. So when I say it allows the vesicle to lose its identity, in Epine, it will break the vesicle. Then the vesicle containing the neurotransmitter, which is the brownish thing, will begin to move across the synapse, the space, and settle on the receptors of neuron B, or the dendrite of neuron B. The neurotransmitter will settle on this area for some reactions to take place. So calcium ions fusing with vesicles for the vesicle to lose its identity, that process is referred to as exocytosis. So the moment then these neurotransmitters settle on the receptors of neuron B, it will cause a reaction. And the reaction it will cause one of the two reactions. It would either cause an excitatory postsynaptic potential. It means that when the neurotransmitter settles on neuron B, it would either cause another action potential to take place. So when it causes an action potential to take place, we call it has led to an excitatory postsynaptic potential. Or sometimes you will say it would excite the other neuron. So when it excites the other neuron again, then we call it excitatory postsynaptic potential. But in situations whereby it prevents neuron B from causing another action potential, or it prevents neuron B from leading to another action potential, then we are saying that it has inhibited neuron B. So we call it inhibitory postsynaptic potential. So get a distinction with excitatory postsynaptic potential. It means that neurotransmitter excited by causing another action potential to happen. Whereas when you say there's an inhibitory postsynaptic potential, it means that neurotransmitter prevented another action potential from taking place. All right. So that'll be all. So my task to you is that I want you yourself to this is not an assignment you'll be submitting to me. it is a personal studies that you yourself can do, because you never know where you might meet it. So I want you to search for some types of neurotransmitters and find out whether they are excitatory or inhibitory. Since you have smartphones, you can easily get them. So we have a neurotransmitter known as dopamine, serotonin, acetylcholine, glutamate, GABA. We have several of them. So find out the various neurotransmitters, about 6 of them, and find out whether they are inhibitory or excitatory. Then again, find out the functions of each of these neurotransmitters. And even possibly find out how these neurotransmitters could lead to certain diseases or mental disorders. So for instance, we have, I'm just giving one example, we have dopamine. Dopamine can either be excitatory or inhibitory neurotransmitter. And one of the core functions of dopamine is that it plays a role in movement. And we normally say that it is implicated in mental disorders or certain diseases because too much of dopamine, when someone has a lot of dopamine in a person's brain, it could lead to a disorder known as schizophrenia. Whereas when another person also have little amount of dopamine in the brain, it could lead to a disorder or disease known as Parkinson's disease. So these are the key things I want you to explore. Find out from, also go through the other neurotransmitters and find out whether it is inhibitory or excitatory, the function, and some mental disorders or disorders it could lead up to, like the way I did with dopamine. Okay. So please, any question? Any question, please? Okay. So if there are no questions, then that'll be all for today.
Hello, sir.
Yes. Hello.
Hello.
Edina. Yeah.
Thank you very much, sir. Please, I want to plead with you if we can get the recording earlier so that we can go to before next weekend. My network didn't help me. So
yeah, and I forgot to say this. Next week I've already set up a quiz. I've already set up a quiz, which it will start, God willing, next Saturday. So it is something you're on the GCUC platform. That's the online website. So as I said last weekend, that if you're not on the website, make sure that you get in touch with management and do it ASAP, because I've already set up an exam for you, a quiz for you. I think it's 20 questions and you are supposed to it's MCQs. And as I said last time, it's not the kind of questions you might see might be different from another test taker. And it covers this, what we just learned is not part. Covers up to the beginning, from the beginning to, I think, is it schools of psychology? Yeah, it covers up to that part, the questions. But the biological explanation that we are doing at the moment is not part. So please make sure that during that time, your Wi-Fi is or your whatever network you are using is really active. And I think I open it up to Monday, so you have ample time to do it. So make sure that you've already been mounted on that GCUC online platform. All right. Okay, Chelsea, your hand is up.
Can you upload this one? Can we get?
Yes, so I'll forward it to your course rep. I'll forward it to Beatrice the link to Beatrice so that you can go to Google Drive and retrieve it. All right, J-Jump.
Yeah. I'd wanted to ask what the lady said, that some of us we join not long. So if the previous topics you taught them, they can send it to us so that we can also go through it before Saturday.
You know, J-Jump, you know your course rep, right?
Yes, please, sir.
So get in touch with Beatrice, because every recordings that I do from the beginning till now, I send her a link for her to for you to go there and retrieve it, the recordings. Okay. So get in touch with Beatrice.
Thank you, sir.
All right.
Thank you, sir.
Okay, so any other thing? Okay, then that'll be all for today. Bye.